Hello, my beautiful geeks. Welcome again to MedGeek TV, the geekiest place on earth. So, today I have a special treat for you. I have a super geek. Steve Ottersberg is one of those brilliant minds where you kind of are like, how does he retain all this information? So, he is a biochemist who has taught at many naturopathic colleges, other colleges, biochemistry. His specialty is really epigenetics as well as cannabinoids. So today we are going to chat about cannabinoids and the influence of CBD and THC on our biochemistry, how it is impacted by our genetics, and the benefits of these cannabinoids on our health. Because this is the field that Steve has been really focusing on for the last few years. He has been working in different laboratories, creating really advanced supplements to really help us get help with cancer, help with different autoimmunities. The amount of information in this man's mind is just mind-blowing. And so I would recommend getting a comfortable seat, maybe getting a tea. You might have to listen to this interview a couple times, but the knowledge will just be pouring out. So enjoy, and we'll see you at the end.
Next level.
And you're recording, right?
I'm recording. Hi, Steve. I'm so excited that you're here with me. How are you doing?
Awesome. How are you doing today?
I'm doing really well. And why I'm so excited to have you here is that everything all over the media is all about CBD and Nutrigenomics and epigenetics. And Steve, you are like the mastermind of epigenetic in relationship to CBD or to just cannabinoids, right? And so I'm super excited about your work and all your research and what you've done. And so can you tell me a little bit, for those of people who don't know what epigenetics is? Could you explain a little bit about what epigenetics is?
Well, by definition, it's a loaded question.
Okay.
Well, epigenetics are the factors that influence gene transcription. So when you think about genetics, you have to think about DNA as the blueprint. Blueprint, you can't live in a blueprint, right?
Right.
So the blueprint has to be transcribed and translated into something that's meaningful and useful. And so, DNA in and of itself, we call that genetics. Like 23andMe would be an example of a genetic test, but that doesn't necessarily tell you if the genes are expressing or not. And what I mean by expressing is that the DNA gene has to be converted into RNA, and the RNA is then converted into a protein structure. And so the protein structure is what we see at real time, but the gene can encode for multiple protein structures to be expressed under different conditions. So here's a great epigenetic trigger that happens in the hooves of caribou. They produce more polyunsaturated fatty acids in their membranes in the wintertime than they do in the summertime. And that acts as a protective agent against freezing because the polyunsaturated fatty acids are liquid at lower temperatures. So an example of a seasonal epigenetic thing in an animal, and we have in our genes, we have the same things. And we have, like today in Durango, Colorado, it's about to snow and it's May 27th. And, we should have epigenetic triggers right now that are telling us to stay awake later and do more and be more active and push our livers and lymph and all that should be happening in the springtime, right?
Right. That is really cool. Yeah, I actually took one of your courses, that whole Nutrigenomic course, and it was fabulous. I would recommend it to anybody watching because you are like the master geek at all of this epigenetic expression. And with the whole thing with cannabinoids, we have endocannabinoid receptors in our bodies. Is that correct?
Correct. But here's the interesting thing about cannabinoid receptors and phytocannabinoids. And so I wanted to make some early on definitions. Number one, when I say phytocannabinoid, that refers to a cannabinoid that's produced by a plant, and a cannabinoid is a chemical type. Does that make sense? So,
Yes.
and so in my language, that has meaning because when I think about cannabinoids, and the history of how the endocannabinoid system was discovered, we discovered phytocannabinoids, THC, first. THC was isolated by Raphael McCoolum in 1963, and since then we knew that we had an effect from this phytocannabinoid. But then over time we learned that effect was because of a receptor system, and we didn't even learn about the CB1 and CB2 receptors until the 90s. Maybe late 80s is when they were first characterized, but they didn't really get broad, their broad understanding didn't hit biochemistry until the 90s, and we're still learning about the molecules we call the endocannabinoids. So we kind of learn this pathway backwards because of the history of marijuana as an illicit, Schedule 1 substance.
Yeah, and you're in Colorado, so that just opened the doorway in terms of being able to test, right? So for a while, you were collaborating with people really understanding CBD, CBG. Can you tell us the different chemical structures like CBD versus CBG?
Yeah, so cannabinoids are produced, and here's the thing that's unique about this plant is that Cannabis sativa is really the only plant that produces the chemical class of molecules known as cannabinoids. There are other plants that produce molecules that bind to cannabinoid receptors, but they're not the same. I call them terpene phenolic compounds, or what cannabinoids are. And so they're produced from molecules known as terpenes, and they form cyclic molecules in enzymes. And there's one master enzyme that produces CBG from terpenes. So CBG is considered the mother of the other cannabinoids. So then there's another family of enzymes that are under epigenetic control. And this family of enzymes are composed, at least what we know now, our current understanding of the plant biochemical pathway is that we have THCA synthase, which is, there's a gene that encodes for the enzyme. The enzyme converts CBGA into THCA. So we have a biochemist where we name things kind of systematically. So we just call it THCA synthase. Then there's a CBDA synthase that also utilizes CBG as a substrate, and it converts CBG into CBDA. And then there's CBCA synthase that converts CBGA into CBCA. So there's this whole family of cannabinoids that are produced because there's a family of enzymes that contributes to their structural diversity.
Now, are these available in hemp as well as marijuana, or only in marijuana?
So here's the plant epigenetics. And
Okay.
so the plant Cannabis sativa has the genome to synthesize all of these enzymes and probably others that we don't know about yet.
Okay.
So the gene contains every cannabis plant, regardless of whether you're going to call it hemp or whether you're going to call it marijuana or cannabis. Cannabis sativa has the genes to produce all of these enzymes all the time.
Ah.
It's the epigenetic triggers that determine which ones are expressed. And so when you look at the evolution of this plant has been associated with the human race for millennia. And it's a very, very anciently conserved plant. The Cannabis sativa plant has not changed significantly since the dinosaurs were on the planet. It's done this for a long time. It has been, and when you look at the evolution of cannabinoid receptors in mammals, it goes to the hydra. I don't even know what a hydra is, but it's a pretty simple organism, right?
Yeah.
So you see a receptor system that's conserved that much within organism relations it means that it's a very old system.
Yeah, you remind me of that Michael Pollan's book, The Botany of Desire.
Mm-hmm.
Yeah, with the relationship with that plant and humans. And so with your work with so you call them cannabinoids, right? So there's many different, right? Okay. With working with cancer or inflammatory or autoimmunity, can you tell me some of your research or work that you've used or educating people on how to use it with cancer?
So there's, number one, cancer is not a single thing.
Yes, right.
You know, you have to think about cancer as a multifactorial disease, and there are as many species of or strains of cannabis as there are types of cancer.
Fascinating.
And there's different reasons why people get cancer. But in general, cancer and just about all other degenerative diseases have a few hallmark relationships, like inflammation. There are certain inflammatory markers that are consistent within just about every disease state. There are prostaglandin inflammation markers that show up in osteoarthritis and cardiovascular disease and in cancer and in Parkinson's, ALS. There are cytokine markers that come up, like TNF alpha. Some of the interleukins, they come up in all of these disease states. And so when you see that there are some commonalities, and this is where, when you look at what the phytocannabinoids do in biological systems, they're in charge of many homeostatic processes, down as core as appetite. So when you think about the importance of something like changing an organism's appetite, it's a pretty core, feature that should come out of their epigenetic expression. But when you think about the relationship between a disease and a plant like this, there's not one answer. There's not one recipe that's going to work for everybody. And this is one of the most important things for people to understand about cannabis therapeutics in general, is you're looking at a multifactorial decision here, because you have, let's just say that there's 10 different cannabinoids potentially in any chemovar that's grown in the United States right now. Notice I'm not using strain anymore. I'm sticking with chemovar.
Okay.
Because strain names are non-descriptive, but a chemovar requires lab data for terpenoids and cannabinoids.
Okay.
So we have a chemical profile versus calling something, bubblegum kush, because you bought bubblegum kush seeds from that dude last year. Remember him?
Yeah. Okay.
So, these, I like to break it down into kind of four simple directions: THC, CBD, myrcene, and limonene.
Okay.
And the reason is because THC, of all of the cannabinoids, probably has the greatest analgesic effect, meaning that it's the greatest pain reliever of all of the family. But it's also psychoactive.
Yes.
And it's a unique psychoactive. It's euphoric, and so people the euphoric effect is too much for some people relative to how much pain relief they get from it.
Okay.
And to give you an idea of its analgesic strength, it's estimated that it's 20 times more effective than aspirin and 8 times more effective than Tylenol, and safer than both. You don't need a liver test to administer THC. It's actually good for the liver in many ways.
Yes.
So if you separate the cannabinoids just simplistically into psychoactives, THC, non-psychoactives, CBD, they both have significant anti-inflammatory effects, both in the sense that they block prostaglandin production, and they're related. There's a lot of crossover between the endocannabinoid system and the prostaglandin system, because they're both built on essential fatty acids.
Okay.
So remember the balance between omega-6s and omega-3s. Absolutely essential for normal prostaglandin production. Remember, too much omega-6 means inflammatory prostaglandin balance, and the omega-3s tend towards anti-inflammatory prostaglandin balance.
Mm-hmm.
So the endocannabinoid system, because it's built on omega-3, essential fatty acids, the balance of omega-3s to omega-6s it directly influences what I call endocannabinoid tone. And endocannabinoid tone, you have to think about it like this. Your body produces the endocannabinoids, anandamide. Anandamide, the chemical name is arachidonoglycerol. Arachidonoglycerol ethanolamide.
Okay.
Got all that?
That's a mouthful.
Or you can call it anandamide. Anandamide was the common name, and it came from the Sanskrit for bliss.
Oh, that's
Yeah,
awesome.
so imagine this, it's a dietary fatty acid that makes you feel blissful.
Yes.
Wouldn't you want the one that makes you feel blissful, or the one that makes you feel kind of numb and blah?
You're right.
So the omega-6 fatty acids in mice have been shown that omega-6 fatty acids directly influence the endocannabinoid-induced neuroplasticity of the brain.
Mm.
So think about that. Too much omega-6, you decrease your neuroplasticity. I don't see that as a positive for anybody anywhere on the planet today.
Right.
We all need to increase our neuroplasticity.
Yes.
This is what gives us problem-solving skills. It gives us coping skills. It gives us, the ability to have tolerance.
And so with the fatty acids, is there a connection between how it's administered? Would it be better absorbed if it was in a fatty form, like a liposome? Do you notice that?
Yeah, absolutely. And this is one of the primary pharmacodynamic parameters of THC that's tricky, is because if you're going to consume it orally, meaning you're taking a tablet or an edible, something that goes into your digestive tract, THC is absorbed at a rate that's proportional to how much fat you ate that day.
Hmm.
So your primary pharmacodynamic parameter of absorption is influenced dramatically by fat intake. So that is a challenge clinically. And then when you consume THC orally, the plant produces delta-9 THC, but when you absorb fats, your fats are absorbed by chylomicrons that are produced from bile salts in your bile. That is the original liposome. This is how your body absorbs fats, is by creating chylomicrons. Chylomicrons are little fat globules that form because they are little, like, detergents that are polar and non-polar, and in an aqueous environment they form little liposomes. That's how all of our fat soluble compounds are absorbed. And it's relatively inefficient, and it requires the release of bile.
Mm-hmm. And so if people have, let's say they don't have a gallbladder, does that affect their ability to absorb it? Yeah.
Yeah, if you don't have a gallbladder, you're going to be deficient in all of your fat soluble nutrients across the board.
That is super fascinating.
They are just passively absorbed.
And then in terms of, we can really geek out now, in terms of epigenetics, when people have certain SNPs, polymorphisms, for people who are watching to know, they're like the dials and switches where how it affects our genetic expression. If they have certain polymorphisms, how does the THC then affect or absorb? Does it have an effect on that, on how it absorbs? Let's say you have COMT, for example.
COMT is not, so here, let's go back to absorption, because the next step in oral absorption is all fats go to the liver and they get sorted in the liver. And so if you have THC in your diet, it goes to your liver, and in the liver, there are two enzymes that go through what's known as phase one, first pass metabolism with delta-9 THC. Cytochrome 2C9 is the primary metabolizer of first pass metabolism of delta-9 into 11-hydroxy-THC. And so now we have to start talking about pharmacokinetics, because now we have a mixture on board of delta-9 THC and 11-hydroxy-THC. And delta-9 THC is relatively predictable. We have typically, if from oral consumption, 30-minute to hour onset of action of delta-9 THC. and a half-life of like 4 to 6 hours. And
Okay.
then there's 11-hydroxy THC, which has an onset of action. We don't know. We think that it has 4 times the binding affinity for the CB1 receptor,
Hmm.
and the kinetics of removal of 11-hydroxy is all over the map. It ranges from 7 to 72 hours. And so this molecule, 11-hydroxy, is converted by cytochrome 2C9, which is a polyvariant gene, meaning I think that we now are up to 8 or 9 different variants of the cytochrome 2C9.
Huh.
So there are ultra slow 2C9 metabolizers that are intermediate slow, kind of slow, just a little bit slow, fast, intermediate fast, and ultra fast variants of this gene. So if you're an ultra fast metabolizer, you're going to convert all of that delta 9 into 11-hydroxy really fast, get really high, probably really too high, and think you're going to die.
Wow.
And the next step is glucuronidation. And so that 11-hydroxy, if you have SNPs in your UGTs and you are a slow glucuronidator, that's a word now, you will have this monster of psychoactivity in your system for up to 72 hours.
Huh.
And this is because of SNPs in the enzyme that affects oral consumption. Remember that if you inhale THC, you don't have first pass metabolism. The delta 9 is, fast onset of action and relatively, predictable duration of activity.
So what I'm understanding is that it's not so much the methylation, it's more of the detox, like the cytochrome P450s.
Yeah,
so
exactly.
you're saying the CYP2C9 is the one that is involved with the metabolism of THC and CBD, like all of the cannabinoids?
CBD, yes, all the cannabinoids are metabolized by cytochromes. But here's the thing about CBD is I'm not going to say it's non-psychoactive because that's incorrect.
Okay.
CBD is psychoactive. It's an anxiolytic.
Yeah. So it helps with anxiety. Okay.
Yes.
Okay.
To me, that's psychoactive, correct?
Yes.
So it's incorrect to say that CBD is non-psychoactive. CBD is non-euphoric.
Yes.
Meaning you can drive a tractor on CBD. You can fly a plane on CBD.
Huh. And so do you feel like it's because the CBD, that anxiolytic factor, has to do with the microbes? What's its effect of on the microbiome?
Well, it goes both ways with your microbiome responds to cannabinoids, and your endocannabinoid system responds to the microbiome.
Got it.
Yeah. And so your cannabinoid intake influences your microbiome, and your microbiome influences the disposition of those molecules.
Wow. That is so fascinating.
Yeah, it's you heard me speak in San Francisco about microbiome cannabinoid. Yes. One of the common things of all of these different mouse studies and cell line studies, there were a couple human studies where they're looking at the DNA diversity of microbe diversity by DNA analysis in the uBiome. I don't remember the other companies that are doing that.
Yes.
There's a very intimate relationship between cannabinoids and your gut function and your microbiome.
Yes.
100%.
It's so fascinating. And so all this teaching that you're doing and lecturing, are you you're not working with the pharmaceutical industry per se, you're working more with doctors at this point?
I'm doing some consulting, but we're currently out looking for investors to fund a big formulation project.
Awesome.
We want to be able to do formulations of unique modes of delivery for different therapeutic classes. and use technology like liposomes to make sure that we're delivering, predictable doses of fat soluble compounds. I mean, that's one of the therapeutic, usefulnesses of liposomes is that they're very useful in delivering, fat soluble compounds that would normally require normal bile function and, all of the other things that influence fat absorption.
Yes. And can you explain to the listeners, I mean, I know what a liposome is, but could you explain what is a liposome?
So a liposome is a little fatty globule that self-assembles. And so these are molecules that occur in nature because of the shape and physical properties of the molecules that make them up. So if you imagine a cell wall is like, a bunch of phospholipids that are lined up with each other, they stack on each other at different orientations based upon what you put in them.
Yeah.
So you can build these little submicroscopic spheres that contain different nutrients, and you can actually engineer the structure of that sphere based on the fatty acids that go into it.
And then it absorbs better into the membrane, correct?
Yeah.
And so I interview everybody in this on my channel that are what I consider health heroes. And you're like a super geek to me, Renaissance man, which is awesome. But I'm sure you've had your own health journey and health struggle. Can you share a little bit about where, all this education that you've done with either epigenetics or endocannabinoids, like how have they helped you with your own health journey?
So it's 2019, correct?
Yes, it is.
So that means I'm 19 years out of surgery for a benign tumor of the synovium in my right knee.
Interesting.
I went into grad school in 1996 as a competitive triathlete and walked out of grad school in 2000 with a cane and thought I was going to be in a wheelchair.
Wow.
And had surgery in 2000, and basically they just removed chunks and chunks of calcifying cartilage. And they had to open up my knee, so I can swim again, I can run again once a year, barefoot on sand only. I can bike, I can ski, but my knee has become my barometer of my biochemistry. I know when there's auto-inflammatory, markers in my body. I know when there's inflammatory markers. I could almost tell you my C-reactive protein based upon how my knee is doing today.
Wow.
And it has been, when I had knee surgery, I think I was still brewing beer. And for me, it was incredibly challenging for me to give up beer because I was such a beer snob that I had to brew my own beer. And after that was probably one of the biggest dietary changes that Naisha and I have done, going down her path of her health, because we're now together for 27 years, married 18 years as of today.
Happy anniversary. Oh, I didn't know. Oh, yay.
And so along the way, she has led the way, and my knee has helped me to follow.
Oh, that's so great.
Yeah.
And you follow a ketogenic diet, is that correct?
Yeah, primarily.
Okay.
Yeah, I mean, for me, for me, it's more about just my health. I spent a lot of time on a now kind of Monsantoized Nebraska farm. And it's heartbreaking for me to see where modern farming is in the United States right now.
Yeah.
At least within my family, and my family's farm started out in the early 1900s, during the teens, farmed by one man with two mules, starting with like 25 acres that wasn't a dowry. It was good to marry women in my family that came with land. But now that my family's farm is 1,000 acres and they don't raise livestock anymore, and it's all GMO and glyphosate, and that, that's, to me, that is where our biggest connection should be moving forward with our health, is figuring out how do we fix that health.
Yes.
The biggest problem in the topsoil today in the Midwest is declining diversity of microbes.
Yes. And so and I'm all for that. I'm a, sustainable ag minor. So that's my love and passion. Do you do regular detoxification protocols in general?
Naisha and I are working, we just did our Great Plains.
Oh, great.
And we were kind of shocked at some of the things that we saw. So yeah, but we've lived through a number of remodels and
Yeah.
old homes, and so, but yeah, we're doing just some kind of phase 2 pushes right now, trying to figure out
Great.
things like how much how much acetylcarnitine versus how much glutathione or N-acetylcysteine.
Mm-hmm.
Like,
yeah,
what I'm doing right now that is just acid. Just trying to see how much acid I need to feel less run down after meals.
Yes, fascinating.
So I'm doing a hydrochloride form of acetylcarnitine.
Yes, and as we say in Chinese medicine, support the liver and the liver will support you, right? So you're supporting your liver in the different phase 2. Yeah, that's awesome. Cool. And so what is the plans for the future? So you're looking for funding to start formulating, right? And are you going to just do CBD or, the cannabinoids, or are you going to look at other things like?
So our goal is we want to be, we're probably going to have 2 different lines. We're probably going to do a physician line and a retail line.
Great.
But, we don't want to be, just thought of as a CBD company or just thought of as a keto company, but we want to be thought of as, part of your toolbox for wellness. And our goal is to be producing, unique formulas that support mitochondrial function, and so that fits in keto, and it fits in primal, it fits in, all of the different camps that people have found themselves in.
Yes.
We want to be like the Swiss Army knife that gets into every camp.
I love it. That's awesome. Yeah. Cool. Steve, this has been so great. So I always ask my guests 3 questions at the end. And so I would love to ask you, if you could have a superpower, what would it be and why?
Organization.
Ooh, tell me why.
Because there's so much information out there right now.
Yeah.
Like when, I get anxiety looking behind the window that you're in on Zoom, because I see like 15 tabs up in one browser window, 20 tabs up in another browser window, 2 PowerPoints up.
Overload. Yeah.
We as a society are bombarded with information, and it's at a level nobody's ever seen this kind of stimulus before in history.
Yes.
And the only way to win is to be organized. And it's because I'm the least organized person you will ever meet in your
life. Well, I encourage you to tap into that power.
If I could get organized and tap my powers, I could rule the world.
You're right. I know you could. All right. So the second question, if you could transform anything in healthcare, whether it's human or planetary, in global healthcare, what would it be?
Everybody has to grow something. I'm not saying that everybody has to grow their own food. Everybody has to grow something that they eat.
Okay.
Everybody has to get dirt underneath their fingernails.
Okay.
And stop calling it dirt, it's soil.
Yes.
You can't grow food in dirt.
Yeah, it's not dirty. It's amazing. Yes.
I love that. Okay.
Saving your fingernails by growing one thing in your lifetime.
Beautiful. That's gorgeous. Okay, and the last question is if you could gift the listener a bit of wisdom or advice to support them in their health, what would that be?
Probably meditate.
Nice. Slowing down, huh?
Yeah, because I know for me that's the hardest. I know I'm probably anybody can do a supplement program or a diet, right?
Yeah,
But to master your mind is a different story.
that is amazing. Coming from from one of the greatest minds I know. Thank you, Steve. It's so awesome to have you on this, and I'm so excited to see where you will be in the future. Where
As are we.
as the next steps. All right. I'll see you soon. Thank you so much.
My pleasure. Take care.
Bye.
Okay, I wasn't kidding. Steve is pretty smart. I hope you enjoyed that interview as much as I did. I know I'm going to have to listen to it a couple times, but his knowledge on cannabinoids, which are just cutting edge, I am so excited for his work and how he's collaborating with his beautiful wife, Dr. Nasha, and I know we will be seeing more of him in the future. I really hope you enjoyed this interview. If you know of any health heroes that have transformed your life, please comment below. I would love to hear from you, and I look forward to seeing you at the next MedGeek TV Health Hero Spotlight.